It is Thursday, June 18th, and you are watching the Biotech Voyager. It's the show that covers what's next in biotech. We synthesize the early stage signals that you should be paying attention to, the companies that are going to be a big deal in potentially a couple of years for acquisition. We're so glad that you've joined us. We're really excited for the show today. We've got a stellar lineup. There's been a bunch of really big moves inside of the space this week. A bunch of different companies have come out of stealth. We have two different interviews with two early stage companies, one that's developing a vaccine for fibrosis, and another one that is backed by Y Combinator that is developing a gene therapy that turns tumors into munitions factories against themselves. So really excited for today. This is going to be a fun show, Jeff.
It is indeed. Lots of things happening. Who is making the vaccine for fibrosis? Base Immune?
Yeah, Base Immune.
Oh, yeah. Okay. That's interesting. I knew that they were making vaccines for like relatively things you wouldn't consider to be vaccinable.
Yeah.
But I had no idea that it was for fibrosis.
Yeah. It's super
okay.
cool.
Very interested to see how they're doing that.
Yeah. Abby on Biologics CDMO. Well, thank you for joining us. We're not quite sure who that is, but a representative, I'm sure, from that company. Thank you for coming. Excellent. All right, Jeff. We'll get right into Campfield. Obviously, they're at the very top here of the leaderboard for the Biotech Voyager. Something that you can go check out over at thebiotechvoyager.com.
Yes, indeed, you can.
Yes, indeed.
Ben's announcer voice.
Yes. But anyway, this is a really fascinating company. So let's just jump right into it.
Let's do it. I gotta fix how this thing looks when I zoom way in. I zoom way in and it just like completely messes this whole thing up. Okay, so yeah, this now, Ben, I actually want to know if you know like why they have the C lowercase.
Yeah, because it's for cyclic AMP, right?
Cyclic AMP. That is correct. So basically, what they've done recently, massive signals. A lot of things kind of all at once emerged from stealth with a $108 million Series A, which is absolutely gigantic for a Series A. Normally you see like 50 million or below. In licensed, so they actually their lead candidate actually is in license from Nusawara Biopharma, and then they initiated a phase 2. I think if I can get this thing on the right signal here, let's see. Yeah, so they initiated their global phase 2 trial. So it's always weird for ulcerative colitis and Crohn's disease, so like kind of like inflammatory bowel disorders. It's always cool when you see a company come out of stealth initiating a phase 2 trial. Like, where were you for your phase 1?
Good gracious.
Okay, so yeah, massive signal here. Number 1 on the leaderboard. So what are they doing? They have a PDE4B small molecule inhibitor for mainly like irritable bowel disorders. So they're called Campfield Therapeutics, and that cyclic AAMP is like the kind of play on words there for camp. Cyclic AAMP is, you can think of it, especially in immune cells, you can think of it as like immunosuppressant, essentially. Cool. It tampers the immune response, and it's got a lot of roles like in T helper cell biology. And we've talked some about T helper cells. They're like the diplomats. They're the Tregs. T helpers they can be immunostimulatory and immunosuppressive. The immunosuppressive T helpers are Tregs. But there's also immunostimulatory T helpers, like Th1 T helpers. Cyclic AAMP inhibits their function. You can think of it like that. PDE4B degrades cyclic AAMP. So if you have a lot of PDE4B activity, you'll have not a lot of cyclic AAMP activity, and then a lot of immunostimulatory stuff happening. So in things like ulcerative colitis and Crohn's disease, these are characterized by highly inflammatory bowel disorders areas. So using a PDE4B selective small molecule inhibitor, you can increase the cyclic AAMP activity and concentration, tampering down local immune responses. Good to go.
Fantastic. And again, cyclic AMP, right, is just kind of like a master secondary signal inside of the intracellular space, right? Like if you've taken any cell biology courses in university, like you've heard of cyclic AMP. That thing is everywhere.
Secondary messengers, like you can think of them as like amplification of signal. Like one activation of one enzyme can create like a flurry or like a cluster of cyclic AMP and amplify signals. So, and PDE4B is known to like degrade those secondary signals.
Right.
So, yeah, being led by Frazier too, massive validation there. $180 million led by Frazier. Let's see who's involved in the actual leadership. Do we know Bill Gerhardt? I wish, what I should do here is put like, where are these people from? Is he from Harvard? I'm pretty sure it's.
Or could we like add in their LinkedIn?
I don't want to put LinkedIn information in here, just like to keep it private. I don't want people to be spamming people, but I could definitely put in like, where do we know you from? I'm pretty sure that this guy is from Harvard.
Nice. So who all invested in this? So we've got like the lead. We've got the lead. We've got the lead here, but who else invested in it? You can see that.
Forbion, huge. Novo, huge. RA Capital, shout out Peter Kolchinsky. Abingworth, huge. Venrock, huge. Deep Track, probably huge, but not as big as like Forbion and Frazier. Right. Forbion, big New York City. RA Capital, big down in. Are they in Boston?
I think they're in Boston.
Yeah.
Massachusetts.
Yeah. Cool. So if you're in biotech, you know RA Capital, you know Forbion, you know Frazier, you probably know Abingworth, you probably know Venrock. Yeah. Big backing here.
Regardless, big validation signals.
Yeah.
Yeah. So there's some serious momentum in behind this.
Yeah. And this is even saying that this, it's obviously a mega round. For those of you who don't know, normally when I see a Series A, anything above like 30 to 50 million is a big Series A. $180 million Series A is ginormous. Yeah. But they're already in phase 2. So they're scaling. Like the technology is basically validated at this point, which, to be honest, is not surprising because a PDE4B small molecule inhibitor, not super risky in terms of like technology validation. They're not pursuing some kind of crazy, LNP delivered bispecific antibody, whatever crazy technology. It's a small molecule inhibitor of a relatively well-characterized enzyme. So phase 2 trial underway.
Fantastic. Excellent. Well, folks, again, we've got a great show planned for today. We're going to be having 2 separate guests later on in the show. We're going to be having Copra Bio on at noon. They are developing some pretty impressive therapeutic applications for turning solid tumors into therapeutic producing factories. And we're also going to be having on the CEO of Base Immune coming up right here at 11:30, focused on a vaccine for fibrosis, of all things. So really excited for that. Please stick around. And folks, if at any point you have any questions or any additional commentary on the different stories or the interviews that we're doing today, feel free to drop a comment and let us know.
Yes. Ask me about my leaderboards.
ask Jeff about his leaderboards. He is very, very proud of them. And I'm very proud of him for making them.
Wow. Thanks, Dad.
Cool.
My dad's proud of me, finally.
Awesome. Okay. Just before we move on to the next story here of Resolution Therapeutics, there was just one thing I wanted to quickly highlight, if that's all right, Jeff, which is this. Okay, perfect. You did it. Just a story that came through. Shout out to Mariam Dineshapur for highlighting this. Unicure is back on the table. They had gone through a rather devastating FDA decision to halt their Huntington's gene therapy. Everyone was kind of surprised, shocked. It didn't really seem to make a whole lot of sense. This seemed like such a promising platform. Huntington's is such a devastating disease. And thankfully, the FDA has reversed that decision. The stock jumped a whole lot on that news. So really exciting to see that this gene therapy is now back in play for potentially going to the market relatively soon. For folks inside of the Huntington disease space. So yeah, just a good piece of news that I figured we should highlight here at the very beginning. Wouldn't you agree, Jeff?
I do indeed.
Excellent. Well done, FDA. We appreciate you allowing this one to go through. Fantastic. Okay, let's go on now to Resolution Therapeutics, Jeff. Does that sound like a plan?
Yes, sir.
Excellent.
It is. Okay, yeah, so let me actually pop out and find out where they are on the leaderboard, because they should be pretty high if they came out of stealth. They're one of the
No, Resolution is not. They didn't come out of stealth.
Okay, well, they're still right here, number 12 on the overall leaderboard.
Okay, perfect.
Pretty good. Let's see where they
Yeah.
are. They're cell therapy. So let's see where they are in cell therapy. Number 2, in vivo. They're just kicking butt lately. So yeah. Okay, resolution therapeutics. Here we go. Now I see some words that I like here. Let's do it. Macrophages.
So
All right,
this
what do we
was
got?
super interesting. Jeff, they're going after cirrhosis. And there's
with
not really a cure
macrophages.
for cirrhosis currently, aside from, sometimes it's kind of associated with certain rather destructive behaviors. But, the damage is often done with cirrhosis, and it's kind of irreversible damage in many ways, shapes, and forms. But this is promising to be a potential therapy, a potential treatment for cirrhosis where one doesn't really exist right now. Not to say that people haven't tried. There have been a lot of different attempts at making therapies or treatments for cirrhosis, and none of them have really taken off. There's a bit of a regulatory graveyard there. But this one from Resolution is heading into phase 1/2, I believe. And the results look really promising. This is going to be a macrophage therapy. And so, Jeff, maybe you could jump in here and explain why macrophages might make sense in the context of liver damage.
Yeah. So macrophages. I'm a cancer biologist. If you're a solid tumor biologist and you hear the word macrophage, you normally get very frightened because they're not good in cancer, because they're very anti-inflammatory, immunosuppressive, right? Now, I'm not a liver guy, so I'm not super familiar with the actual etiology and physiology, pathology of cirrhosis, but I'm gonna guess it has to do with inflammation in the liver. Is that a safe bet? Let's see.
I believe so.
I would think cirrhosis.
Perfect.
Late stage. I can hear you. Yeah.
Okay, perfect. Awesome.
Fibrosis caused by long-term liver diseases or injuries. Okay, so it's fibrotic deposits in the liver, which also
could this be a potential application with base immune later, because they're also going after fibrosis for scarring.
It depends on.
Interesting.
It depends on what their vaccine does.
Yes.
Now, I'm going to go on a limb. If you're in the comments, please tell me how wrong I am. We're going to have a recurring section on the show called Tell Jeff Why He Is Wrong. Because I'm always just like, here, I'm a biologist and here's what I think's happening, but I'm probably wrong. But it makes sense in my head. So here we go. Macrophages are phagocytes. That is the truth. And they gobble up stuff. Okay. I'm going to guess they can probably gobble up these fibrous things that are causing this fibrosis mediated liver damage. So sending in a bunch of hungry hippo macrophages to eat up and chew up these fibrous networks is likely the mechanism of action here.
Gotcha.
Macrophages and like monocytes, myeloid cells as a whole, are underutilized, in my opinion. I think one of the biggest problems with a myeloid cell therapy is it's tough. They're tough to make. They normally have very short half-lives. It's hard to make these things and get them into the body. And I think I saw that this is autologous, which makes it even crazier to me. Let's see. Yeah, they're engineering an autologous macrophages with pro-regenerative properties to address the chronic inflammation and fibrosis underlying organ-damaging diseases such as cirrhosis, right? Yeah. Its lead asset, RTX-001, is engineered with IL-10 and MMP9 mRNA. We can get to that to amplify anti-inflammatory and anti-fibrotic activity. Well, there it is. Okay, so this is exactly what it is. Okay, yeah. In end-stage liver disease cirrhosis. Okay, so IL-10, it's an immunosuppressive cytokine. IL-10 in tumors, very, very bad. You don't want it. And macrophages can secrete a bunch of it, right? And then, so obviously, like this will be anti-inflammatory, immunosuppressive to tamp down on inflammation. And then MMP9 is a matrometallic protease, which these are secreted, and then they can break down extracellular matrix, including probably fibrotic structures. So anti-inflammatory, anti-fibrotic, you got it right there.
Nice.
These macrophages can get right in there, and they're very good at like squirming their way deep into fibrous networks, which you'll have to do if you want to get in there because it's characterized by fibrotic networks. And then they'll kind of sit there and secrete, and then they can also chew things up. So they're very good at like tissue healing, tamping down on inflammation and like healing tissue. But in cancer, for example, again, I'm a cancer guy. You don't want the tumor to be healed, right? You want that tissue to be destroyed. So
Yes.
a lot of macrophages in tumors is bad because they promote the healing and regeneration of the tumor. But in a chronic liver disease like this, you would want a lot of macrophages in there to promote tissue healing.
Cool.
Okay, now cue the section. Tell Jeff how wrong he is.
Overall, I would agree. I think that makes sense. It's it's super interesting. They are at a phase 1/2, right? So they're trying to figure out like what's the tolerated dose, how, you know, like what's the safety profile look like. But yeah, the only other thing I will flag with the story, it says in particular it's focusing on decompensated liver cirrhosis. I don't know if that's different. I'm not sure what the decompensated part means, but regardless,
yeah, I don't know. Decompensated.
If anyone else knows what decompensated liver cirrhosis is, feel free to drop it in the comments.
You know who will know what it is?
Pardon?
Google will know what it is.
Google.
Decompensated liver cirrhosis is an advanced, life-threatening stage of liver disease where the liver can no longer perform its vital functions.
Oh, okay. So it's basically like late stage cirrhosis.
Yeah, this is like the liver's no longer even functioning. It's bad.
That makes sense, right? It makes sense to kind of go after the most life-threatening form of disease, right?
Indeed.
Cool.
Yep, well, and they're currently the lead sponsor in recruiting. Yep, this is all from clinicaltrials.gov. Phase 1, RTX-001 in end-stage liver disease, cirrhosis, liver cirrhosis, decompensated. Perfect.
Nice.
All right, moving on.
Nice. One thing just quickly, Jeff. So about the leaderboard here, there are filters.
There is indeed filters.
Okay. So,
let's say,
for example, if I wanted to look at cell therapies inside of neurology, what would that look like?
That would look like this.
Well, you can just do that.
Super, super simple. And this is just like the companies that have active like heat signatures. Okay. But if you wanted to see all the companies just like in alphabetical order, you could go to the company board. But here is all the companies that have shown a recent activity in cell therapy in neurology. And here's Trix. We talked about them the other day, about 2 weeks ago, maybe. So here is their signal. We talked about they presented clinical data in multiple sclerosis. This is a Treg cell or TR1. Yeah, Treg TR1s in multiple sclerosis. So yeah, cell therapy in neurology. What else you got? Let's do
that's awesome.
Cell therapy utilizing AI. 3 companies. Who you got? Anaka, talked about them on Tuesday. Waypoint Bio, talked about them last week. Lunai, what did Lunai do? They got a big signal over here. What happened here? Let's see. They raised some money. There you go. 20 million to, yeah. Okay. So preferred equity, whatever. Okay. That's fine. So yeah, pretty cool, man.
Nice. Right on.
Okay. So who's next? What do we got?
Excellent. All right. So once again, folks, we've got 2 different guests coming up here. At the halfway point, we're going to be talking with Kevin Walton, the CEO at Base Immune. They're developing vaccines for scarring. If you can believe that. Super excited to dive into their approach. And then also Andrew Bartinsky just raised $9.1 million over at Copra Bio to go after solid tumors. And they've developed a gene therapy application that allows you to basically, instead of turning tumors into snitches, you're turning them into a munitions factory that's going to blow up in their face. So looking forward to that. Excellent. All right. So moving on here very quickly. So I guess what's up next here is Vidana.
Oh yeah, okay. So they're pretty high because they came out of stealth. Here we go, number 8. Bam.
Yes.
Emerged from stealth. 46 million Series A. See, this is more what you typically see between like 25 to like $50 million Series A's. So high end Series A, Westlake and Canaan. Let's see what they're doing. This is the one where I just like don't understand any of this. I saw this and I was like, oh, GPCRs. And it's like, nope, you're dyslexic. It's not GPCRs. So CGRP. I don't know, man. I'm not a migraine guy. Not really super sure. So what do you got for me? Anything? Did you look into this?
Not a whole lot. But again, there was basically just like 4 different companies that emerged from stealth. So we wanted to cover all of them. So
Yep.
yeah, we got a couple minutes here. Let's dive into what this one looks like. So they emerged from stealth. They're focusing on, what was it, migraines, you said?
Yeah, I think it's they got a couple antibody programs. PACAP, which again, I see this and I'm like, oh, PARP. Nope, you're dyslexic. Nope. Not GPCRs, not PARP. So yeah, this is like very outside my wheelhouse. Now it's saying subcutaneous for migraine prevention, which that's super interesting. So I also want to emphasize the fact that we're getting a lot of subcutaneous antibody therapeutics lately. Johnson & Johnson came out with, I don't know what the actual therapeutic was. I want to say it's a PD-1. I don't think it is though. But there's something happened with antibody therapeutic design where you can now have them as subcutaneous formulations, which is a big deal because normally you need to do infusions. So having it subcutaneous, like a vaccine injection sort of thing,
right,
of an antibody therapeutic. It's crazy. That's actually like revolutionary. So that's super interesting. Subcutaneous delivery of migraine prevention, which you would need to have. If you have an antibody therapeutic for migraines, you couldn't do an infusion. It just wouldn't be like the burden on the patient the juice wouldn't be worth the squeeze. You know what I'm saying? Like if I had a migraine and I had to go to the hospital and get an infusion, I just wouldn't go. I'd just deal with it. Right. Yeah. But a subcutaneous injection that I can do myself at home, no brainer. Yeah.
No kidding. Especially if it's prevention. So you're not even waiting for it to show up. You can just be like, I know I'm susceptible to this. Got a big day coming up.
Right.
Gonna shoot myself.
Shoot myself up. Yeah.
Shoot myself up just a little bit.
Yeah.
with that good antibody.
Yeah. Yeah, no, nice. But it's still super cool. And here's the really interesting thing that I want to emphasize, Ben. If it wasn't for the Voyager, we'd have no idea this company even existed. You know what I'm saying? Like we'd have no idea to even focus on this. Yeah. Most likely, right? But here we are talking about subcutaneous antibodies.
Burn down the silos. Just one big barn.
Exactly.
Yeah. The big barn of biotech. Cool.
So, can we pretty please do multivalent biotherapies next?
We certainly can.
I know a lot about this stuff.
Okay.
Okay, so multivalent biotherapies. First and foremost, couple signals here. Emerged from stealth, $27 million Series A. Again, right in that good range. Relatively lower end, of a Series A. I think I have an idea as to why it's not super large. You can also, I won't go down this road yet, actually. The size of the Series A can actually give you some idea of the complexity of the therapeutic. A massive Series A normally means like you're late phase clinical, or you have a very complicated therapeutic that needs a lot of money. $26 million, $27 million Series A, that's actually a pretty good sign. It means that this thing is probably not super like monetarily complex.
Complicated.
Yeah.
Well, for example, right, like when we're going back and looking at Campfield, right, they are going straight to phase 2. So,
they're scaling. They're
scaling.
Yeah. Series A's pre-phase 1 normally is like technology validation. And if you get like an $80 million Series A for a preclinical company, that normally isn't a good sign. That's a hot take from Jeff, but I see that and I'm like, ooh, that's weird. Like, why are we giving almost $100 million to a preclinical company for a Series A raise? Right. They must need it for some reason, which is not good. But this is much more in line with what you'd expect. And what they are doing, Benjamin, is they have peptide drug conjugates, which you probably know of, antibody drug conjugates.
Yes.
They're very, very common, right? Now, which means that if multivalent biotherapies is making peptide drug conjugates, it's likely a direct competitor to antibody drug conjugates, which is like bold, right? ADCs is a massive industry, especially in oncology. Like coming after ADCs is a big deal. So the question is, what is a peptide drug conjugate? Well, it's a peptide conjugated to a drug. There you go.
What's the advantage there though? Like, is this something that's going to be a bit more systemic versus antibodies being more specific?
Not Not quite. So the difference between a peptide drug conjugate and an antibody drug conjugate, there are many differences, and it's probably better to go to the limitations of antibody drug conjugates first to then describe how PDCs overcome those. The two big ones are size of the therapeutic, right? I guess there's three big ones. Size. Antibodies are massive and they can't diffuse very well. They can't get into tissue very well. So when it comes to treating solid tumors, you don't want to have problems diffusing through tissue. You want to get deep into the tumor. ADCs typically suffer from that. Two, they got a very long half-life, which could be good theoretically to have a long half-life on your therapeutic, but it gives more room for toxicities. If you have these ADCs just kind of floating around for a long time, you open up more room for toxicities. And then number three, antibodies they take a long time to develop and they're expensive.
Right.
Peptide drug conjugates promise to overcome all three of those. You can control, they're basically like a custom peptide, right? So the reason you use an antibody drug conjugate is because the antibody can bind to a desired target. Right? HER2, for example, right? And HER2. If you can create, all an antibody is a protein that binds to something. So now that we have these advanced protein synthesis, like AI models and stuff, we can just create our own custom binders.
Right.
So we don't need antibodies anymore. Antibodies are good because you can basically just like inoculate some animal and get an antibody out, right? Which is like simple to do, like scientifically, I guess, or like theoretically, but it's expensive, long, it's tough. It's harder to design a custom binder, or at least it was in the past. Now that we have a lot of these protein folding algorithms and like AI-based protein generation programs, we can begin to actually make our own custom binders, and we can give them the properties that we want. We aren't beholden to nature, kind of. So we can have them have desired half-lives. We can make them small or make them large. We can have them do different things and then conjugate a drug to it, and you have a custom peptide binder drug conjugate, right? And I think that what multivalent is doing is they have like multi-specific binders. So this picture isn't the best. It's kind of dopey, but you can imagine that like this is a peptide, okay?
Right.
And it's binding to two different targets, and it's got a drug conjugated to it, right? That's what they're doing, so that you can have them bind. Whereas with like if you wanted to have a multi-specific drug or multi-specific antibody drug conjugate, you would need to design like a bispecific or trispecific antibody thing. With the custom peptides, you just design a multi-specific peptide or a tri-specific or quadra-specific peptide, tetraspecific peptide to bind to all kinds of different targets, conjugate a drug to it, and you're good to go. We're going to see more of this in other areas, such as like radioligand therapy.
Right.
And also I think like universal CAR T cell therapies, where you have like a soluble binder to a target that binds to a CAR receptor, and then you can have like multi-specific adapters for CAR T cells. Look into Avencell, for example, Avencell. Companies like that. You're going to see more and more of this kind of stuff because it's getting easier and easier to design, synthesize, and scale custom peptides.
Nice. And I guess just even with like machine learning and AI, right? It's become that much easier to design. Yeah,
exactly.
So that's kind of what's held this back from being something that's been done historically. It's literally just been too resource intensive to go down that far of a custom path.
Yeah, you need to rely on nature. You need to basically say like we're just gonna screen a bunch of stuff and like figure out what can bind here, and we don't really know why or how or whatever, but we know it's binding to this thing. There we go. We have a binder. But now we can do like rational design using AI, learning from different types of interactions. Like we can do things deliberately now, which changes the game.
Interesting. And then even with regards to like custom biologics in this way, right? Do you see this as potentially a path? I know I harp on this every once in a while. Like, do you think we might get close to a what's the best way to say this? Like a driver's license for custom biologics? Or
maybe this would be the kind. I think that in that regard, maybe gene therapy might come first.
Okay.
Like custom gene therapies, like maybe like for example, like cancer vaccines that you can. That gets complicated. Gotcha. I don't think that the first stop on that road probably isn't custom peptide therapeutics. It's probably somewhere in the area of gene therapy.
Gotcha. Very good. Well, ladies and gentlemen, our first guest has arrived today. Kevin Walton is in the green room, so we're going to be bringing him on just momentarily here. So again, Kevin is working on some pretty cool stuff. Let's just take a quick look at Base Immune here. Preclinical, working on some really cool applications for vaccines inside the fibrosis space. So let's bring him on. With no further ado, let's bring him into the studio. Kevin, welcome to the show.
Hey, how are you?
We're doing well. Good to see you.
Thanks for having me.
Glad to. All right, so I guess just like right off the base, can you give us a little bit of background on Base Immune? What's kind of the big problem you're going after here?
Yeah, Base Immune, we're tackling fibrosis with our active immunotherapy approach. That's based on a computational platform where we can multiplex epitopes on an antigen. And we're going after idiopathic pulmonary fibrosis, or IPF, first. It's a devastating disease where scar tissue progressively replaces healthy lung tissue. It's hundreds of thousands of patients globally. The life expectancy upon diagnosis is 3 to 5 years, so it's a devastating disease. There's been decades of research, and there's only been 3 drugs approved that only slow the slope of decline. Nothing halts or reverses it. And then there's been a lot of failures in the clinic. And I think that's kind of where we think we can be quite differentiated. We think a lot of the failures are due to everything going after a single target. But the issue with the biology of fibrosis is that it's inherently a network-driven, redundant disease. And so our computational platform allows us to go after multiple pathways with the same active immunotherapy.
Super cool. So when I think of it's an anti-fibrosis vaccine.
Yes.
When I think of vaccines, I think of my immune system killing something. What is the immune system killing in this case?
Yeah, so I'd say it's a therapeutic vaccine, so it's a little bit different. It essentially uses your immune system as the generator of antibodies, polyclonal antibodies for multiple targets from the same vaccine to go and then block and prevent the progression of fibrosis. And so the goal is...
Okay, that makes way more sense.
The ultimate goal, right, would be to halt the progression, but kind of like if you look at it as fibrosis is very much analogous to cancer. And so if you look at it in that respect, if you're able to significantly slow the decline, you're then going to add many, many years of life. Right now, the 3 drugs out there, it's not a huge meaningful improvement. And the tolerability issues are such that many patients drop after less than a year on treatment.
Okay, so are the targets pathologically associated targets, or are they things that we would have tolerance to?
One of the things we have to do with our synthetic epitopes is to break tolerance. So we need to make them different enough to break tolerance, but similar enough that you still...
Cross-reactivity.
Exactly.
Yeah. Okay. So okay. Now it makes way more sense. There are a lot of companies who are doing this is much more elegant than what other companies do. Like I know companies like Vironexus, for example, they just use an AAV derived or AAV mediated like gene therapy to have hepatocytes produce a bispecific antibody for cancer therapy, for example. Where the therapy is meant to have your body producing some kind of therapeutic antibody, right? This is kind of the same thing essentially, but this is much more elegant and immunologically like nuanced.
Yeah, it's similar to, you have like the Moderna and Biotechs of the world doing the cancer therapeutics, cancer vaccines. You actually have quite a few vaccines going after Alzheimer's. You also have some for things like other autoimmune disorders like lupus. You have some for cardiovascular as well. And so I think the modality, or maybe not modality, but the approach is not new. But I think what we're able to do is we're able to do this in a way where you can go after multiple pathways. And especially for something like fibrosis, where it is pathway driven and shutting down one pathway, other pathways compensate for it. I think that's where we can see hopefully a much more transformative impact on patients.
So that's super elegant. I love it. Is there a reasonable risk of toxicities. Since you're breaking that tolerance, could you get some kind of like extended cross reactivity that damages tissue?
Yeah, I think the biggest safety question, right, is right now in development, a lot of the candidates in like phase 2 are monoclonal antibodies. And so you infuse the patient with monoclonal antibodies, there's a half-life, and then they go away. Now the bad part is, right, if you want to keep having a impact on the patient, you have to do chronic infusions. Whereas for our approach, you would likely have to get boosters, but it'd be much less frequent, and it's a booster, not going into the hospital, getting hooked up to an IV and having that infusion. The thing though is how do you turn it off?
Yeah.
Because there's been ways shown that you can. I think also the way that your body is just innately operates is that it doesn't keep producing it when it's gone. If you think about other therapeutic vaccines, like in Alzheimer's, I think you have some phase 2 assets, and it shows that there isn't necessarily a safety issue. You also show that if you look at some of the monoclonal antibodies safety issues, getting those infusions, you don't have a lot of necessarily bad side effects elsewhere in the body. But that is something that we will have to obviously monitor closely.
Sure. Yep.
Yep.
So with previous approaches that have gone after this disease and they haven't been really all that successful, is it partially due to the fact that they're going after just one pathway, and so you kind of just get rerouting around it?
Exactly. Everything right now is really single target, and so the belief, right, is, and if you maybe take a step back from an evolutionary standpoint, it makes sense. If your lung gets damaged, you want to make sure your body can find a way to heal it. Now, fibrosis, pulmonary fibrosis, is really just the dysregulation of that mechanism where it overheals and it keeps producing that scar tissue, which takes up the healthy tissue. And so taking one pathway, one target, the other pathways will compensate because it's been dysregulated. So your body keeps thinking we need to produce more to heal.
Okay. Interesting. And then I guess, like, just in terms of like the early data that you guys have seen, like what kind of got you excited here? Like what was the thesis where you're like, oh shoot, we've got to make this into a company. This is something that patients need.
Yeah, so it's actually interesting. So base immune's been around a little bit longer, and so we started off looking at infectious diseases based on our platform, and so we were able to generate some compelling data in coronavirus, where you can actually make one vaccine that can work across multiple variants. So instead of saying what's the next variant, you can actually have something that lasts longer because of the ability to go after multiple targets. We also had some compelling data separately in malaria. And then we wanted to pivot into chronic diseases. And when we were looking at the wide landscape of chronic diseases, there was, I'd say, 5 or 6 that really boiled to the top. And we chose fibrosis over the other ones specifically because I think it is really custom fit for the disease and the disease biology. And I think you have a number of programs that have worked a little bit in phase 2. And so you can see, okay, if you go after that target, there is an efficacy signal that does help impact fibrosis. However, right, it likely isn't doing enough on its own. And so by looking at kind of the validated targets, and when I say validated, I mean that you have some type of efficacy signal. We think that if you take enough of those and put them together in our approach, you can have a much more transformative impact. The issue of infusing kind of cocktail monoclonal antibodies is you're going to have a lot more tox issues, and you're going to have different issues there that we don't necessarily encounter with ours. And so far, what we've been able to show is in silico, we still express the antigens that we're trying to. They bind to clinical mAbs, so we can make sure the epitope's intact. When immunized, they're breaking tolerance, so they're generating antibodies, and that's an important factor based on the early comment from Jeff. And then we also have shown that we can reduce collagen in some bleomycin models. And so, working currently through some in vivo models as well, but so far everything's been very encouraging in the last 9 months.
Nice.
Sounds good. So I'm very curious, and tell me to be quiet if I'm approaching any kind of proprietary information, but I'm curious, you mentioned that the biology of fibrosis was such that it was amenable to this approach. Does it have to do with the actual, like the ability to use this multiplexing discovery engine to find like candidates that will work immunologically and break tolerance? Is it kind of like in that realm, or it's something different?
I'd say it's a little bit different. So we do have some ways to find novel targets, and that can kind of work across diseases. Right now for fibrosis, we're specifically focusing on more known targets that have shown some type of efficacy signal in phase 2. And that's because we're taking a lot of technological risk. And so how do you balance that with the biological risk?
Sure. Yeah.
And I think the technology is especially custom fit for fibrosis just because of needing to go after multiple pathways that would be really hard to do in other approaches and combination therapies. And I think it's the ability to kind of, the one thing we've done a lot of work on is looking at the different pathway drivers of fibrosis, what targets are kind of incorporate or under each one, and how can we make sure that we ultimately get a therapy that's going to hit at least 2 or 3 of those.
Yeah. So I don't want to divert the conversation too much, but this is it's approaching one of the things that I have a very passionate opinion about, which is cancer vaccines. I'm gonna assume that the biology is amenable also in fibrosis more so than it is in cancer, because the fibrotic like tissue is not so aggressively immunosuppressive and difficult to have an immune response there. Is there something there, or am I mistaken?
Yeah, I think the tumor microenvironment is going to obviously be very different than like the extracellular matrix environment from fibrosis. I think the other thing is that you also on tumors, and I'm not a biologist like you are, so I will defer to you on that. But I think within oncology, like I know the cancer vaccines, they're really looking at a lot of the neoantigens that are specific to the tumor, whereas fibrosis is really an inflammation. And if you think about it, it's an inflammatory type disease where it's your own cells, right? It's not neoantigens that are unique. And so I think that is a big difference, is how do you go after kind of self targets, proteins.
Got it. Yeah, one of the things, I mean, I have a lot of opinions about the cancer vaccine space. One of them is that we basically treat the cancer vaccines like prophylactic vaccines, and it's just not the same type of physiology. Like the biology just is not the same between like protecting against chickenpox and killing an active tumor. And the technology doesn't seem to really appreciate that as much as it should. But it seems like fibrosis would be a different beast altogether, and the problems with cancer vaccines wouldn't really apply to a fibrosis vaccine.
Yeah, no, I think that's great. I look at more of how would you go after, say, a neurodegenerative disease, cardiovascular disease, pain, things like that, where it's more kind of looking at like self proteins versus the tumor microenvironment.
So,
Nice.
so I want to pull on one thread that you just mentioned earlier, right? You're going through looking at these different chronic diseases. 5 or 6 surface to the top. Obviously, fibrosis is the top candidate, the largest unmet need. But I would be curious, like what other disease indications surfaced in that initial discovery phase?
Yeah, I think one that I'll share 2 that I got pretty excited about as well. One of them is pain. And I think it's a very similar thing. There's a lot of drivers of pain.
Pain vaccine. Oh my gosh. Okay.
What in the world?
But, I think the thing I would challenge there is think of it more of an active immunotherapy. So not necessarily you're taking it as a preventative prophylactic, but it's more therapeutic, where if you were to get chronic migraines, you would take this, and then that would help in terms of the intensity, the severity, but also the frequency.
But imagine the marketing opportunity I mean, even if the biology is technically not like this, imagine the marketing opportunity of providing a vaccine against pain. That would just kill in public.
It would be, you would have to isolate what are the main drivers for like migraines in terms of just like the biology,
of course,
and then develop something that goes around that. But I think, I have a headache, I can go and take, acetaminophen or ibuprofen, and that helps with the headache. But that's not necessarily like the blockers of the causes of it. And it's also not a migraine. If I have a migraine, that's a different cause. And so if you can isolate certain routes and then go after those, that's huge. And it's typically going to be multiple routes, right? And same for cardiovascular diseases. So you have a lot of patients who are taking multiple drugs to help essentially your cardiovascular health. How can you actually design one that's going to be much more powerful and doesn't have, you're not taking an assortment or a cocktail of
Right.
small molecule drugs and pills just to try to like maintain a healthy heart. And especially with, I think that, the last few years, there's been a lot of research on new targets, and you hear a lot more like, LP little a, things like that.
Yeah, I think the combination of that aspect where we're just like learning more about biology as time goes on, right? Combined with AI and the ability to like generate these new, like you guys, I mean, like, a multiplex vaccine that breaks tolerance, like that would be basically impossible 15 years ago, right? But now with these really powerful computational models and things like that, I'm not super sure how yours works, but it becomes much more feasible to do these things.
Yeah. I unfortunately I can't tell you how it works, but it's I
won't ask.
By machine learning. Yeah, well, but you're right. I mean, we're able to down select from hundreds of thousands very quickly using our machine learning program.
Yeah.
Excellent. So we actually have a comment here just from a bio briefcase here. You made antigen-specific T cells. Great. Can they infiltrate a tumor? Can they overcome immunosuppression? Can they avoid anergy? It's just kind of in the context here of the problem of cancer vaccines. They're also saying that they love the content. Well, thank you at bio briefcase. We appreciate you coming. So I guess maybe
we know him.
Do we? Perfect. I'm blanking. My brain is mush. My child was up at 4 o'clock this morning. But yeah, that's an interesting.
Yes. So we have a T cell. So our platform, we have a B cell approach that's like a VLP protein, and then a T cell approach that's mRNA LNP. For the mRNA LNP, I haven't talked much about that approach, but that's more of a pain fibrosis approach where was listening to a show earlier. It's a little similar to resolution therapeutics, but it's different because it's not cell therapy. It's still active immunotherapy or a therapeutic vaccination. And what it's doing is it's essentially allowing T cells to go and destroy the fibrotic tissue. So it's a little bit analogous to, I think, the macrophage approach, but also different. We've not done anything in oncology, but I think that there is the potential for the technology to be applied there. But, like Jeff was saying earlier, there is also differences in terms of just disease biology. And I think that, we are not oncology experts at the company. And so we're more immunology experts. And so that's something that we've stayed away from on purpose.
Yeah, oncology is a different beast, especially when it comes to vaccines. It's a tough. I would wait before you guys even have any kind of inclination to think about doing oncology vaccines. Just wait until we get some positive results from Moderna or whatever is going on over there.
Probably later
otherwise,
this year they'll have their first big pivotal readout. It'll be interesting. I think there is positivity, but how much of that is? Yeah, it's tough to dissect out because it's also a combination with Keytruda. So
true. Yeah, Keytruda is 80% of the response. So,
but an amplifier and a meaningful improvement in life expectancy is also a great thing. But
I think you're going to have some kind of like adjuvant on top of the adjuvant when it comes to those things. Keytruda being one of those things, including like cytokines and all kinds of stuff. So it's a whole like.
It's also very different too, because there you have to analyze the tumor for the neoantigens. You can't just say here's the 3 or 4 big targets or pathways and do it for everyone. It's a unique, individualized therapy for every patient, which is a very different ballgame.
Yeah, you would need to have like a similar system to yours, but have it be like basically real time.
Exactly. Yeah. But we have shown in malaria a T cell approach that's been fantastic. It's performed much better than most of the academic models because of the ability to go after the targets and be much more specific. So the T cell approach is something that is promising, but that's kind of more of our second program after the IPF.
Yeah,
fantastic.
I like the antibody approach a lot. That's like super. There's so many companies doing that with like autologous B cells and things like that. So to have it be like a vaccine approach, that's really interesting. So
yes,
we're excited.
Yeah,
nice. All right, so Kevin, I guess just the last question here would be, what's next for Base Immune? What should people be watching out for?
Yeah, we're generating some in vivo efficacy data right now in mice, and then we'll look to use that to go after a multi-target approach to essentially design and optimize the lead drug candidate. Hope to get to proof of biology and IND enabling in the next 18 months, and then
nice.
Go from there.
Excellent. All right, so there is a customary question we try to ask each guest just at the end here, which would be, what's being slept on in biotech that's going to be really big in a couple of years? Ideally a pithy response, 30 seconds or less.
Sure. I'm biased because we're producing active immunotherapies, but I think
Everyone
really
is.
the immune system as a drug manufacturing platform that you can utilize. I think the biology that we have inside of ourselves is amazing with the immune system. It's an incredibly sophisticated drug factory. And so it's really, how do you exploit that for going after these diseases?
So you mentioned that you're a bunch of immunology nerds. That was great. So you mentioned that you're not oncology guys, but you're immunology guys. I'm also an immunology nerd, and I totally agree with that sentiment. Utilizing nature has gifted us with this incredibly complex system that is designed to heal our body. We should be using it to heal our body. And we're only now being able to uncover, or I guess exploit those things with the advances in technology. But I think that we're only scratching the surface, and I love that answer. It's great.
Excellent.
Thanks.
Well, Kevin, we really appreciate your time. We know we went a little bit over here. Thank you so much for coming, and we look forward to seeing your progress into the future here. Thanks so much.
Thanks so much for having me. Thank you, guys.
You are most welcome. All right. Thanks so much for coming, Kevin. Right on.
Sweet. And honestly, Ben, so I didn't know how their vaccine worked. Now that I do, it actually is like kind of similar. Now it's definitely not actually similar at all, but it's in the same like thread as Copra, who we're going to have in a little bit. Utilizing I'm not super sure if Base Immune is like a nucleic acid based vaccine or if it's a peptide vaccine, but like you can think of it as we're going to make our body produce and secrete the drug that we want to make. That's what they're doing. And Copra does the same thing. Siren Bio does the same thing. Vironexus does the same thing. A lot of companies are now saying, hey, why are we making the drugs outside of the body with like random cell lines when we can just give our body the instructions to make the drug itself?
Right.
That's like the general thesis of a lot of these new companies.
Super cool. Yeah. I'm really interested to see as well. Like, man, they got to lean into that. Like a vaccine for pain, that would crush.
That would really crush. That would absolutely crush
so much.
Like,
I'll take a vaccine for pain.
Yeah. Although, would you want to have no pain ever? Isn't pain good?
No. There's all sorts of horror stories about that. Absolutely.
Yeah.
Not.
Yeah.
But if it's somewhat similar to like the migraine story that we were talking about earlier too, right? Like, if you're struggling with migraines and you have a way to prevent that, go for it.
Yeah. Yeah. So I'm actually curious to know, I'm being a total dork right now. I want to know the oncology gene therapy, 70 companies.
Oh. There's 70 companies inside of oncology and gene therapy.
So here's Vironexus that I mentioned. Siren's got to be in here somewhere. There's Siren. Now they're counting all of the in vivo CAR T cell companies as gene therapy for oncology. But like you get companies like Vironexus. So I mentioned Vironexus. What they do is they have an AAV based gene like therapy that causes liver cells to produce T cell engagers, right? So it's our body creating an antibody therapeutic. This is the exact same thing that Base Immune's doing. Just they're using a vaccine approach to do like custom epitope antigen targeting antibodies. Super cool. Yeah.
Nifty. Cool. All right. So folks, just in a couple minutes here, we're going to be having our final guest of the day on. Andrew Bartinsky just raised $9.1 million to go after turning cancer tumors into drug-producing factories.
Cancer tumors. I love how you say cancer tumors. Just say tumors. They're all cancerous.
Fair enough. Solid tumors into cancer in cancer therapeutic factories. Second here, we've got a comment here. Guys, I got a question for you all. Bio briefcase, hit us with it. What's your question?
Ben, oh my gosh,
like
his name is slipping my mind. We met with him like 2 or 3 weeks ago. He's the guy up in Canada who, come on, we know.
Sorry. I'm really sorry. Again, my brain is mush. We should know your name.
No, we know him.
Hit us with your question. So if that comes through here, okay. So just a second here. We'll be talking about Spot Biosystems in a second, but Bio Briefcase has a question. And
Rob Mould.
Rob Mould. Oh, Rob, right on. Hi, Rob.
Yes, it's Rob.
Fellow Northerner. Love it. You're almost a Northerner, Jeff.
I'm basically in Canada.
Yeah.
Not now, but I'm from there.
Yeah.
Buffalo. Go Bills. Go Sabres. They suck. Copra. Oh yeah, he's UCSF and Y Combinator. Perfect.
Yes.
In vivo tumor editing.
This is something I wanna focus on. We're seeing Y Combinator, I would say, starting to focus a little bit more on biotech over the last couple of years, I would say.
I wonder if this can search. No, it can't do it. Okay, that's fine.
Oh, here's the question. There's going to be a barrage of AI-powered developed drugs coming down the pipeline. Were there any standout companies that cut through the noise and hype that you guys are stoked for? Yes, for me, in silico medicines, all the way. They are the basic. Come on, no, but they've just got a really powerful pipeline. Alex, the CEO, has been in AI forever, way before it was cool. They've just got such a really powerful, validated platform for finding AI design, for designing molecules with AI, generating the initial efficacy data, and then kind of packaging it up and selling it off to big pharma. It's such a powerful model that just kind of keeps things going. And Alex is always, all right, Rob, I feel attacked. Calm down.
What's your favorite food? Pizza. Yeah.
But again, they just have a really powerful model. I got to meet with them. I got to meet with him rather, and he's just an interesting LinkedIn following. You never know if he's going to have like some really powerful take on AI, or if it's just going to be pictures of him shirtless, ripping chin-ups in his hotel room.
Facts.
It's always a roll of the dice with Alex.
Mm-hmm. So the one company we talked about them. This is like off the top of my head. Kiragen. Here's what I was looking for. Kiragen Bio. Super interesting company. Aaron. Using AI to discover how to overcome the tumor microenvironment in cancer. Right. So like there's just like infinite possibilities in the various types of tumor and microenvironments. They use an AI based like learning model that will help them using like preclinical data and things like that, figure out the best like multiplex gene editing for their CAR T cells. Really cool. We also talked about Anaka the other day. Anaka's got a really interesting machine learning like APC derived TCR discovery platform for TCR T cell therapies. So look into Anaka. Anaka is out in Sweden. And then one more, Antiverse. Antiverse, companies like Antiverse. Oh gosh, I got to fix that. People doing like AI-based drug discovery for classically hard to target receptors and other kinds of like transcription factors. So Antiverse in particular is doing GPCRs, and ion channels. But there are a lot of companies who are using like AI-based discovery models to identify targets for things like GPCRs, ion channels, transcription factors, things like that. Antiverse being one of them.
Right on. Get the biggest loan you can and buy calls on in silico medicine, Ben. This financial advice is not brought to you by the Biotech Voyager.
Yeah, we do not provide any kind of investment advice.
All of those different disclaimers, they apply retroactively to this conversation.
Exactly.
Excellent. Yes. So, ladies and gentlemen, Andrew Bartinsky has joined the green room. We'll be bringing him on in just a moment. Again, very excited for the approach. They were just able to raise $9.1 million to go after turning solid tumors into cancer therapeutic producing factories. So we look forward to learning a little bit more about that with him. So with no further ado, let's bring him on. Andrew, welcome to the Biotech Voyager.
Wow, what an introduction. I can't believe it. The drum roll and everything.
You have no idea. That's a very long time coming. We've been trying to get the soundboard to work for weeks.
Yeah,
Ben's doing a good job.
I'm finally earning my keep. Andrew, how have you been? I know back when we did the Bio to Bedside, we had you on just to kind of have like a Y Combinator dedicated episode.
Yep.
And we had you guys on, and then also Leah Labs on. We had Leah Labs on last week, was it, Jeff, or the week before?
It's
They're
Velocity
spinning out a new
Therapeutics
company,
now.
so it's really cool
Oh,
to see.
that's so funny that we're like
Yeah.
all on the same cadence. Amazing.
Mm-hmm.
Excellent. So yeah, bring us up to date since then. So before, you guys were in the very early stages, trying to generate data, and now you've got the money to take this to the clinic. How have things been?
Got like a couple of detectives. The image that you used to like promo me coming on the show, I think, is like the first findable image of Alex and I together ever.
Nice.
So that was fantastic. So yeah, back in 2024, we started the company. Based on work that was done at UCSF. And so this is work that Alex did during his postdoc. And what we were able to do is develop basically a very tumor-targeted vector that allows us to deliver genetic payloads. We're sort of calling this concept in vivo tumor engineering, right? So, in vivo CAR T, so hot right now. But what if you could think about approaches that could deliver the same outcomes for a solid tumor? So instead of modifying immune cells, you could go in and modify tumor cells. And instead of making them make, targeting ligands that you hope are present on the solid tumor, what if you could deliver therapeutic proteins? And so what we've been able to do is deliver a very potent IL-15 superagonist. And so that's sort of the first program that we're launching with. And then we spent the past 2 years, putting together the heart and soul of the company, putting together the core technology, starting to do development, and raising the capital that'll help us sort of get this first product into the clinic. And so now, it's great to be able to announce that, and we're really excited that hopefully early next year we'll be dosing patients and trying to make a difference in one of the most challenging solid tumors, which is glioblastoma.
Nice. Okay, so first,
people getting going into clinic. Yes.
keep our studio audience under control, Ben. I have questions to ask. Jeez. So it's in glioblastoma. How are you getting the thing in there? That's the first question.
Yeah. So the great thing about working with neurosurgeons and neuro-oncologists is that they're great at delivery to tumors that are located within the CNS. And so for GBM, which is usually a single locus disease, direct into tumoral injection works very well. And for basically the majority of viral therapies that have been delivered for GBM, they've all been local delivery. So we're using a catheter-based delivery system and going right after the tumor.
Nice. And then, so there are a few other companies who are doing kind of similar things. One of the ones that I'm thinking of is Siren Biotechnology. They're not doing IL-15. They're doing, I think, IL-12. But what kind of vector are you using? Is it self moderating? I guess is kind of a dumb word to use, but you wouldn't want to have extended inflammation in the brain, right? So so
there's a couple of people who have thought about various versions of this approach, and the sort of goal is how can you try and get persistent therapeutic delivery in a tumor that's very inaccessible, right? So if you were trying to do like systemic delivery, has been very challenging in this indication. Because you have to cross the blood-brain barrier. So if you're going to be local, how do you get something to stay there, and how do you get it to be produced locally? Obviously, there's been an incredible amount of CNS development work for AAV. And so that's been the vector of choice for some other companies in the space. Trogenics is one. I think they're the one that's doing the IL-12. And then Siren is the other. And so I think there's obviously decision-making when it comes down to selecting your therapeutic payload. And some of that just comes down to how you think about translatability. And so for us selecting IL-15, what we thought was really interesting about that construct is it is cross-reactive in both rodents and humans. So when we're actually doing our preclinical studies, we're actually using the same exact construct that we're going to be using in patients. And so we think that for translatability, having that be a one-to-one understanding of how this vector is going to perform is much more helpful than having to design like a rodent construct and then hoping once you rejigger things inside of your vector that's not going to have some subtle but maybe important impacts on what's going on in patients. And then it's just about thinking about the biology of what you think is going to be the best in terms of the signaling that you're trying to do with an inflammatory pathway. And so for us, we like IL-15 a lot. It's got a lot of really de-risked biology, and there's an approved IL-15 product in bladder cancer. So we think like taking the ensemble of data together, IL-15 just felt like a very good choice for us.
Yeah. So are you guys following along with Immunity Bio? The Anktiva is like kind of kicking butt.
Right. Yes, yes, very, very closely following that product, of course, and like hope for it to have continued success.
Exactly. Yeah. Good news for you guys.
Yeah.
Excellent. So I guess with regards to other approaches that have been on the market, right, for something like glioblastoma, why have other approaches failed here?
Yeah, so I think the thesis that we have for GBM and sort of more broadly for solid tumors is really come down to delivery. So as I was mentioning before, issues with getting therapy across the blood-brain barrier is really difficult. And so trying to solve this local production of therapy where it's most needed, which is basically in the site of the disease, we think that's one of the major barriers that we're trying to overcome. Beyond that, we're largely trying to flip cold tumors to hot tumors. And so trying to take this approach of being a little bit inflammatory, we think is very interesting and could be one of the unlocks for some of these difficult to treat solid tumors. There's been a couple of recent targeted therapies that have done quite well in DMG. And so there is, for people who possess those mutations, there's been some work moving forward. But when we're talking about really poorly mutated tumors that don't really have a wide variety of targets that are accessible on most patients, trying to go after something that's a little bit mutation agnostic, we think is also really interesting.
Yeah. So I'm wondering here, I'm a GBM guy, kind of. I did a postdoc in CAR T cells and GBM. And the big thing the biggest problem with that is like macrophage biology. They're like half the tumor is macrophages. So I'm curious if the choice of IL-15 had to do with like macrophage biology as well, or if it was just like, nope, super agonist the crap out of those things, get T cell activity, NK cell activity, and just like, shoot them with inflammation, basically.
Yeah, we were primarily looking at the immune side of things. And so when we were doing this study, and so it was just published in Nature Communications, which is the culmination of many years of work. It was great for Alex and the team at UCSF. And so we did a bunch of work trying to characterize the tumors post-administration of the treatment, looking for signs that were actually biologically following the sort of model that we're putting forward. Because in my opinion, right, rodent models, like the survival benefits that we're able to demonstrate are exceptional. But I think that rodent models have not really translated very well in terms of, curing cancer in a mouse doesn't necessarily result in a cure in a human. But what I think mouse models are beneficial for doing is trying to validate that your biology is functioning in the way in which you're proposing. And so what we were trying to look for is signs that we're actually able to remodel the TME, and we are actually driving in T cell and NK cell populations. So through both flow cytometry and RNA-seq, you're able to show really strong evidence that that's occurring. So that's another piece of sort of building the foundation of why we think this therapy is so promising.
So I think I found the paper here. I'm going to read this thing later. So non-lytic viral immunotherapy induces long-term glioblastoma. Yeah, perfect.
Yeah, okay, Put it right next to your bed. It'll help you. You'll do a great
Put
job.
it under my pillow.
You'll never do better. Yeah.
Yes. Perfect.
the biotech fairy comes along and leaves you $5.
Yeah. And your IND approval under your pillow.
Exactly. Yeah, I found that like passive diffusion is generally the best way to ingest scientific knowledge. Yeah.
Yes,
exactly. Yes, from paper to brain.
Yes. Excellent. Okay, so I'd be curious, Andrew, just to go back, and folks, for those who are watching again, thank you for coming. If you have any questions for Andrew, feel free to put them in the chat, and we will get to them post-haste. But just, I guess, for you, Andrew, again, this is something I'm really curious to double click on, which is, again, you took an approach that I'm very interested in, right? In that you went the Y Combinator approach, which hasn't been super common inside of kind of the wider biotech space. Why was that, and kind of where do you see this going in terms of like Y Combinator's interest in biotech in the future?
Yeah, for sure. So I mean, I think we're in like a sort of Cambrian explosion moment for founder-led biotech because the cost structure is just completely flipped. So I think like much in the same way for tech, that like the advent of like the cloud storage, like AWS and Google Cloud, et cetera, made it easy for basically anyone to spin up a tech company. I think now that the cost of getting to clinical data has gone down by a factor of 10x, I think that'll just enable a lot more founder-led biotech companies to get to an inflection point where they can then raise capital in terms that are more similar to a tech company. And so I think the reason why this path was not common in the past is because perhaps you were reliant on like a large incumbent fund to sort of escort you through the early stages because the requirements were so high. But in the current environment, again, with the cash that we've been able to raise, we'll be able to get human data. And I think that's about probably like 50 to 75% less than it would cost, even 5 years ago, what people were thinking about. And this is like a cell and gene therapy. This is a very complicated biological product. And so I think that when those cost structures flip, these models become more attractive. And so I think for founders who would like to be in the driver's seat for their companies, going through the Y Combinator route is a good way to do that. And I think the network supports that. And you can get enough capital now to get to these inflection points where you can really make a difference.
That I love to hear that because this is a little bit of a self plug, but we are starting this platform called the Biotech Voyager that's going to be explicitly dedicated to tracking these early stage companies. And I think like I really like the tech industry where it's like everyone and their mother on Twitter has got their own tech company. I'd love to see that in biotech where it's just like we have all these really new, young, gritty, like millennial-y kind of Gen X biotech companies that are like some guy. Ben, you were the one kind of blowing this horn 6 months ago. Some guy in his basement with an AI program designs some therapeutic, gives it to his dog.
Yeah, the dog story.
Right?
Yeah.
And it's like,
it's crazy.
Okay, I might be a little bit more on board with this like new agey like biotech synthesis.
Yeah. And the other story that's been really interesting to follow is Sid from GitHub.
Yes, GitLab.
Where he basically really aggressively took on ownership of his cancer care, went and developed a bunch of personalized treatments in a way that most people wouldn't have thought was possible, 10 years ago because of the tools that are available now.
Yeah. It could be
pros are coming for biotech. I've been telling you.
Yeah, a part of me is like not envying the FDA. Because it's going to be a regulatory nightmare. I think it's going to be like we're going to have so many. It's cool on one hand, but immediately like the biologist in me is like that's dangerous. Bah. So what are your thoughts on that?
So I mean, 100%. So I think that doing this in sort of a risk appropriate manner is going to be the path forward. And so the way that I think we could start to think about things is look at what people are doing elsewhere, right? So we don't have to reinvent the wheel. We're not it's not like there's no biotech activity going on outside of the US. So we've actually been able to benefit from sort of surveilling what people are doing in other jurisdictions and seeing what works and being able to learn from what doesn't. And so places that I think are good examples of how you can do this in sort of a risk appropriate fashion would be Australia, right? Australia is a great example. That's been a pathway that people have been doing for probably now the past 10 to 15 years. And they just have a very different approach to early phase clinical studies than the US does, where again, patient numbers are on the smaller side. And so if you're thinking about, the risk of a cardiovascular drug that's going to be in 2 million people, like, that once you're making a decision on whether or not that level of exposure to the population is going to occur, I think you need to be very risk averse. But when you're dealing with, serious life-threatening diseases in, 1 to 10 patients, I think you should sort of right size the requirements to get to that stage to see if this is worth pursuing further. Because if it's not, you can just think of the hundreds of millions to billions of dollars that go into programs that ultimately fail relatively early in clinical development. And so the less expensive we can make those failures, the more potential successes we can have. And so beyond Australia, obviously the other example that now everyone's doing is China. And it's very similar in the way that they have sort of decentralized the review process for early stage work. And again, I think sort of taken a risk benefit framework to small early proof of concept that I think is really interesting for trying to sort of drive through biotech innovation in a way where it hopefully no longer costs us like $2 billion per drug to approval.
Yeah, and I'm thinking that this is a little bit of a long shot and might sound like completely stupid. I don't know. Here we go. I think that with the maturation of AI-based analysis, we might be able to get to a point where we can like have relatively confident and robust like benchmarking very early to say like, here's the signatures that we're looking for in a pre-IND therapeutic that will predict phase 2 efficacy. Maybe not now, maybe not in 5 years, but maybe like 10 years, we'll be able to get like enough data that was generated with the purpose that says like, we can now be predicting things very early and maybe cut things off before we have to figure them out in the real world, quote unquote.
Totally. Yeah. Part of me is very optimistic for that. Part of me also wonders how many potentially good drugs were canned at the rodent stage.
Sure.
Because the rodent biology is not representative, and they were just like, "Oops, we'll never touch that again." But it's just like maybe there was something there that we. I don't imagine.
There are some companies. We featured one, I think last week, where people are kind of like buying the shelved drugs from pharma that may have had like maybe lackluster phase 1s or phase 2s. And they're now saying like, let's give that a relook. So that's kind of happening, but I think, yes, like just like scouring over all of the negative results from like, years past could get really interesting.
Yeah, I think my like axe to grind is I think that sometimes we get really infatuated with like large numbers and screens. Like a lot of times people get really, it's like we did 500 million variants, we did 1 billion variants, and like what actually matters is the space that you're sampling. And so like looking at very high signal areas with smaller things, so like looking at drugs that have already had human experience and trying to find where they might fit is probably a much more efficient approach to getting winners than trying to sort of boil the ocean when there's really only like 2 or 3 grains of sand that are interesting, right?
Sure. Yeah, I think that when I was in grad school, they had a whole initiative of like drug repurposing, where they would do just that, where they would take the repertoire of approved therapeutics and try and see like, hey, does this heart drug work in glioblastoma, right? Or like, does this oncology drug work in heart disease, right? So there's also, I think too that the massive fishing experiments might not be as difficult to arrive at a conclusion because of AI these days as well. But in the past, it's just kind of like we can just take all the things that our chemists can produce, throw it in these cell lines, and see if anything sticks.
We have robots now. Let's just run, 96
we do 35 million.
Yay.
Right.
All right, so there are a couple comments just want to get through here quickly. Rob, you might have your question cut off here. Might be a silly question for you, Andrew, but is turning a GBM tumor hot? I'm not sure if that question got cut off.
So that's like immunologically going from a cold tumor to a hot tumor. So like, yes, that's what we're trying to do. So GBM is traditionally thought of as a cold tumor, as are most solid tumors, in that their microenvironment is basically immunoexclusionary. So if you were to like take a biopsy and look at all the cells and see which ones are immune cells versus which ones are tumor cells, you'd find like only very few T cells there. And so what we're trying to do with the cytokine that we put on board is to dramatically increase the presence of those immune cells within the tumor and then drive them to start killing these aberrantly mutated cells that they encounter.
Yeah, it's a very strange term. It's immunologically hot, right? People think, oh my gosh, you don't want to turn it like the temperature up, right? In your brain.
I just think of the Zoolander quote, GBM so hot right now.
Exactly. Yeah.
Chris Spivey also has a comment. Sorry, wrong one here. Oops. Here we go. Chris. Yes, patient stratification also falls in the same bucket. I think he's going back to just the cost of.
Yeah, and I think knowing the biology better is one of the things. The first time that I really appreciated it was with Avenzo Therapeutics. The key to your phase 1 and phase 2 success is patient stratification, especially phase 2. Knowing how your drug works and knowing which patients it won't work in is one of the most important things. But it requires you to know how your drug works, which is usually like not as well thought through as you'd hope.
Yeah, well, I think it was, what is it, like the Keytruda versus Opdivo, like one of them stratified better in terms of PD-L1 expression, and that's why Keytruda won.
Yeah, makes sense.
So it can come down to these early understandings of mechanism and trying to make sure that you're trying to enrich your population for responders, and then you can get more and more understanding as you get to a larger and larger data set.
Yeah. The Avenzo Therapeutics one was they have a CDK2 inhibitor and they stratified for high cyclin E expression, which would make it sensitive to CDK2 inhibition. So it's like knowing how your drug works, finding the patients, getting the right biomarker, and that will be easier with our increased ability to do like tumor biopsy based approach, like analysis and AI, things like single cell and like high throughput IHCs. And then knowing how your drug works is like the other half of that. So
totally.
Okay, Rob's question did get a bit cut off, and it was just with he was going to ask: are there risks in terms of like the pressure or inflammation of a productive immune response in the brain? Oh, that's a good question.
Yeah, so this is a really good question, and when you're thinking about these treatments, right? You want to induce an inflammatory response because that's almost a signal of potential efficacy. But then, of course, within the CNS, there's potential risk to that. And so when designing the clinical protocol for these types of drugs, you always include sort of an inflammatory control schedule, where if there are signs of clinically risky inflammation occurring, there are a number of things that you can do to tamp it back down. And so, as a part of any reasonable clinical development plan and any reasonable clinical trial, you think about these potential, on-target, off-target effects of your drug, and then you think about if-then, basically, in trying to design control mechanisms to maintain the safety for the patient.
Love it.
Yeah, Siren Biotechnology, they have like an AAV-based one. So it's like as the cancer cells die, you're going to get less expression of interferon beta. That's like, there's got to be some kind of like safety system built into the therapeutic, or you're just going to get, brain damage.
Right. But yeah, you can dose with things like steroids and other things to tamp back inflation.
True. Yeah.
Super cool. Okay, so I guess just in terms of in terms of like the data that you've been able to generate so far, like what would you say was the really compelling piece that I guess made prompted this investment to go forward into the clinic?
Yeah, so the thing that first caught my eye, and I think has been largely the driver of people's belief in the technology, has just been the efficacy signals in multiple really aggressive GBM models in rodents. And so these are very, very fast growing. They grow substantially faster than what you would see in a human. And so if you're able to control those tumors to the point where we get basically complete regressions in some models, that's a really strong signal that you're making a big difference. So I think that's one. People like survival benefit, and that's sort of a good metric. And then the second one for me, which I think is most important, because I'm always just thinking about the human biology is going to actually rule the day. Like we don't get approved based on rodent data. So it's how thoroughly have you vetted the way that your therapy works, and how much have you validated that it's doing what you say it's going to do. And so for us, really looking at the detailed mechanistics of like, does the virus selectively infect the tumor? Does it replicate within the tumor? Does it produce functional payload? Does that functional payload induce the correct biological response? And the fact that we were able to validate every step of that pathway in exquisite detail was, for me, what made me want to take the leap and go and like, let's actually push this forward.
So I have such a nerdy question for you.
Yes.
It has to do with the data from the paper. I'm looking at some of these of like the mouse data. I see TU2449 re-challenge data.
Yes.
So when I see re-challenge, normally I think of like things like vaccines. When you guys do the re-challenge, do you put the tumor in the same spot? That's the first question. And do you perfect? Okay, good. Yeah. And do you give the drug again, or is it like a?
We do not. Yeah. So this is like yeah. So again, as a way of sort of validating that we're doing an immunologic mechanism and not some virus response that's being triggered, locally or whatever. That was why we did the re-challenge experiment. And so seeing tumor rejection, I think it was like 65 days after the first tumor, shows that you're really creating like durable immune memory within the immune cell population of the animal.
Yeah, and the big thing with GBM is like recurrence. You can,
correct.
It's like almost you need to be able to, you need to be able to have your thing have an abscopal effect, or you're done. Like you're going to have metastasis in the brain and recurrence. So you've got to be able to re-challenge and get a good response in different locations. So really good to see that.
Yeah, it's that durability. Yeah,
and it's a straight line, 100%, no mouse deaths to report. Which again, we can cure cancer in a mouse. We've done it over and over again. but getting it into humans. But it's I mean, what else do you do, right? You've got to
exactly. It's the best. Yeah,
then,
you work with the tools. Yeah, you work with
Right.
the tools that you have. And again, for me, it's about answering mechanistic questions, and like survival is a sexy figure, and like great, we have incredible survival data, like so good for people who care about that. But what I care about is like, is it doing the biological things that we say it's going to do?
Do we see like CD8s
right? So,
and like antigen specific?
Exactly.
Yeah.
Exactly.
Nice.
Exactly.
Yeah. No, it looks really nice. I'm gonna dig into this later, and maybe I'll have some more questions to ask Alex.
Yeah, it was very strategic for me to not share it before we came on.
Nice.
Yeah, get the friendly questions now, and I'll get the hard ones later.
Great. Yeah. No, I'll be nice. I promise.
Yeah, so I guess just in terms of next steps here for the company. So I think I said right, you're planning on doing your clinical testing in China.
Yes.
I would be curious to, like, first of all, we can jump into that, like what are next directions for the company, but I would be curious to just kind of jump into that as well, right? With like, especially for someone in your position, right? You mentioned earlier, right, the cost of developing these therapies and like evaluation and whatnot has reduced by tenfold. I'd be curious to get your thoughts as well on just like what role China is playing in all this and kind of where you see them slotting in the future. There's lots of debate going on right now on biotech Twitter about, whether or not this is kind of like spelling the end of US biotech dominance, or if there's going to be, different parts of the industry just getting outsourced to different parts of the world. I guess maybe let's jump into that, and then we can end off with directions of where you guys are planning on going next. But I'd be curious to get your thoughts as someone who's in the weeds of this right now.
Yeah, so I mean, I think still innovation is coming from the US, and you're seeing the most novel technologies coming out of the US. Funding for that high risk research has been dramatically in flux, I would say, for the past couple of years. And so if that funding is not there, I do think we will see dominance in innovation. And so that'd be the first thing, is that if you don't invest in innovation locally, you won't be able to maintain it. And the people that are innovative are some of the most mobile people on the planet. And so they will go where there is capitals to support them. So I think if we want to support innovation, we need to invest in it locally. I think that the development side of things is and should be just kind of like an open marketplace. Like these are in many ways commodity services. And so we should be able to go to the lowest cost provider, because once again, that'll allow us to try to develop more drugs. And the more drugs we put into the pipeline, the more successful drugs we get at the other side. I think that once you get to approved drugs, then it again becomes, I think, interesting and important to invest in domestic production and to support that. Because I think these are like critical pieces of the economy and the healthcare infrastructure. And so you would want to be able to not be reliant necessarily on, other parties for all of them, or for all the raw materials. And so like, that's where I think I land on, is like, you need to support innovation. I don't think there's a better like cultural tolerance for risk anywhere else than the US.
Yeah.
But then I think once it gets to development, that should just be a commodity, and we should want to push the prices of those tasks as low as possible, because then it gives us better benefit for approved drugs. And then, finally, once it gets to approval time, we should want to have a robust manufacturing infrastructure for late stage and commercial products, so that we can support them at home and abroad. So that's what I think about that. And then in terms of how we think about the context of doing drug development in that world, again, our job is to, as quickly and as cost effectively as possible, understand the human performance of our technology. And so that's the mission that I'm on right now. And then as soon as I have that understanding, obviously the US is going to be where we're doing the majority of our development work for phase 1 to phase 3. And so, right now it's about taking technical risk off the table. And then it's about, doing the best sort of commercial and, where's the best clinical trial infrastructure in the world for mid to late stage development.
Yeah, that all makes perfect sense. Yeah.
Yeah. And it's crazy. I don't know why it's like such a, like, oh my God, we have to scream at each other about this. It's like, no, let's just kind of all be reasonable.
Yeah. And so the one thing that I might want to push back a little bit on is, like, if America is the one that's fueling the innovation, shouldn't it be the people in America who potentially benefit from like the clinical trial participation?
I mean, I think they do, right? The drugs that are developed through this process are first available in the United States usually, right?
So if you do a clinical trial, if you do your phase 1 and phase 2 in China, though,
Yeah.
then like the patients in America aren't eligible for that.
I mean, that's like 200 patients, right?
Okay, that's almost,
yeah. So if like, and like it's 200 patients in an unproven drug. So I think that what I, the people that sign consent waivers and actually participate in clinical research are incredibly brave and are like hugely important for society. But I don't think that we should think about those patients as being like the access strategy that we care about, right? The access strategy we care about is where do approved medicines launch first. And that's in the US. And so the second point beyond that, right? So like who's going to be the first 10,000 people to get access? They're probably going to be in the US. The second question is then what happens when those drugs go off patent and then more and more people in the US and worldwide are able to do it. And so I think that it's a system, again, that rewards risk taking for breakthrough medicines. I think there are potentially bad actors that are trying to leverage some of the economics in ways that are, financially beneficial and not really participating in the innovation part of the business. And we should probably think about how to fix that. But I think in order to reward the risk and just facts of how many drugs fail along the way, and you need to be able to support those on the back end. I think we need a system that is able to financially support that, and also still giving U.S. patients the first access to some of the best medications that are coming through.
Yeah. And the thing about the clinical trial patients don't benefit in the U.S. It's like you're right. It's like 50 people. So shut up. We're gonna get this thing. We're gonna prove it. We're gonna prove it, and then once we know it's good, we can offer it widely.
Exactly. And like the faster we get through trials, the more people will be able to do. So like if I'm stuck trying to enroll a 200 patients study in the US and enrollment is going slow, that seems way more problematic for potential patients that would benefit than if I got approved 2 years sooner and opened the floodgates to the masses 2 years sooner.
Exactly.
And so yeah.
Clip that. That's a yeah. Perfect.
I don't know, you just have to take a step back from like the screaming and yelling that occurs on the internet, and then just try and like go to first principles and think about like what is the world that you want to live in and how do we make that happen, right?
Yeah,
so just
exactly.
Put those pieces together. Yeah.
Gotcha. Okay, we have a second question here from Chris Spivey. We love Chris over here at the Biotech Voyager. Shout out, Chris. Andrew, when first broaching your approach to new contacts, what is their most skeptical question or most difficult aspect for them to grasp? Chris coming in with some hot questions.
It's a good question. I think generally gene therapy is harder for the layperson to grok and understand. And so like getting people to think about like how it works, we usually spend a lot of time talking about how it works and just like what are the steps that occur. Because chemotherapy, antibodies, they're sort of just like very understandable, like lock and key kind of stuff. People can understand that pretty quickly. It's been around for 100 years for drugs and pushing 50, 75 for biologics. And so it's just for gene therapy when you have to talk about sort of all of these multi-step processes that occur, getting people to understand.
Well, you see this thing called transcription and gene expression.
Okay. Yeah, it's like the mitochondria is the powerhouse of the cell. It's like, you just like have to.
my wife's one biology fact that she knows what the mitochondria is, and I'm like, but there's this like RNA polymerase 3 does this, and it's like
right. And so and so like how up and down the stack can you go with people to get them to understand what are the necessary and sufficient things that are happening to make your drug work. And so having that conversation is usually where things start, and once we can get them across the sort of value of understanding on that, then it comes into like how fast can we get this to patients and
I'm sure that the mRNA based like COVID vaccine didn't really help with public sentiment. There's probably a lot of mixed feelings about like, oh, it's like the COVID vaccine. It's like, well, not really, but okay.
Yeah, I mean, you can't control what things end up like bubbling up to the public consciousness. And so then you just try, we like to use I Am Legend as a sort of touchstone, right?
Okay. With Will Smith, that one.
Yeah. Well, and it was a cancer vaccine that originally created the zombies.
Oh, really?
That was great. Yeah, it's great.
Nice.
It's great.
It seems to be mildly in line with my gripes over cancer vaccines, i.e., they'll never work to treat cancer. They might do something else, but it's not going to treat cancer.
Yeah.
All right, Andrew, I know we're a little bit over time. We appreciate you coming
Yes.
on. We do ask, there's one question, just we try to get like a pithy take from you, 30 seconds or less.
Oh, for sure.
And the question is, what's slept on right now in biotech that people should be paying attention to?
I think China is still slept on. I think like as much hype as there, like you just have to go there. Like everybody who works in this space should just go there, like see it on the ground, interact with the community. I think that US companies have just assumed that they would be dominant and the fastest option to do things. And it's like going to Shenzhen for electronics. Like you just need to kind of see it to understand what we need to be able to do if we want to be competitive here. Yeah.
Excellent. On that note, thank you so much, Andrew, for coming on. We know we're a little bit over.
Yep.
Appreciate you moving around your calendar. We're looking forward to following you guys into the future. Cheers.
Yep. Talk soon, guys. Bye-bye.
Thanks, man.
Right on. Man, so, you know, as the Canadian coming in here, so I know kind of like the US versus China debate is always interesting. And then I'm kind of almost in the sidelines because Canada isn't really in the same playing field as those two. In a lot of ways, I like the take where people are like, actually, the US needs China because to an extent, you need a strong competitor in order to kind of cut through the kind of like the sitting on the laurels maybe a little bit, right? Where it's like, hey, now that there is kind of like an active competitor on the globe and you're not just like the dominant player anymore, and you kind of maybe get a little bit lazy and processes get a bit more bureaucratic and things kind of slow down because you're so far in the lead. If there's someone else there who's also running alongside you that's kind of forcing you to keep pace and run a little bit faster, as much as it kind of hurts in the short term, it might actually be a good thing long term. So that's my thought on that whole side of things. Fight you can grump at me if you like, Jeff.
I think that my whole sentiment is, can't we all just get along? It sounds like so like 1960s.
Sure, but it's also a competition. It is a race. There's so much money to be made, and you know everyone. I
understand like that arms race, like competition can breed what is it, innovation
Excellence.
Or whatever. But yeah, but also I think that there is this like underlying competition in things outside of biotech that poison the interactions.
Sure.
I think he's right. Like, why can't we just rely on, like, you know, for example, like your clothes are probably made in China.
Right.
This thing's probably made in China, like my little fidget thing. Like, it doesn't. Why does that have to then immediately? I'm not going to go down this road. Actually, it gets political. But I think in terms of the actual biotech stuff, like I do agree that we should take the most efficient path to drug approval. Why play politics with, you know, a clinical trial for your bispecific cancer antibody, right? At the same time, I think that there is some kind of like, it is more nuanced than that, especially when in America. I think that, yeah, we won't get into the I'm not a political commentator by any means. So,
right on. Okay. Spot Biosystems, Jeff. I believe this is the final of the four different companies that emerged from stealth this week.
Yes. Okay, so the big thing here, as you can see from the picture, maybe, is this is a gene therapy for Duchenne's.
Right on. And
added to the list.
EVs.
That's the difference.
Yeah.
So the delivery vehicle is extracellular vesicles and not like AAV or LNP or the normal things you would expect, mainly AAV. So yeah, Spot Biosystems develops engineered extracellular vesicles for non-viral, low immunogenicity mRNA delivery, addressing the immunogenicity and targeting limitations of viral vectors and lipid nanoparticles. Its core tech uses tissue targeting EVs, endogenously loaded with therapeutic mRNA cargo, and blah, 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 blah. The rest is history, right? The big thing with AAVs, Ben, you can probably attest to this better than I can, is immunogenicity and the lack of redosing potential.
Yeah.
I think EVs is like a direct answer to the immunogenicity problem. They're less immunogenic. They may be easier to produce. I don't know. You got to get them from cells. But yeah, this is a mRNA delivery play.
Cool.
Spot Biosystems. Now, the actual signals here is, yes, they came out of stealth. So come on, give me so launched from stealth, 40 million venture financing. Maybe, I don't even, is that Series A? I don't know. $40 million.
That seems worthy of an applause.
Yay, Spot. And then they also, I think they dosed their first pediatric DMD patient. So again, Duchenne's muscular dystrophy, very, very common rare disease for gene therapy, express the full length or some kind of truncated version of dystrophin. I'm wondering also payload capacity. I'm wondering if you can get like the full length dystrophin, whereas with AAVs, you typically do some kind of truncated version. Not really sure.
Yeah, that would be a big thing, because right now, if you want to use AAV for dystrophin, you've got to deliver basically microdystrophin, so like a truncated version of the protein. Dystrophin is one of the largest proteins in the body, full stop. And so that has been a challenge with AAV in the past. And so using EVs instead, right, extracellular vesicles, could be a very promising way to get around that. The one challenge that I have heard with EVs, and maybe they can fix this, is it's just really hard to get like a consistent product. And so maybe they've got some sort of manufacturing process that helps with that. I've heard some people say like, hey, it's not really that bad with EVs. It's basically like getting a bowl of Skittles. Yeah, there are different colors, but they are all Skittles, as opposed to AAV being like, well, I only want the red Skittles.
Yeah, the problem is like, it isn't just different colors. It's normally like the size is different. That's the biggest thing. The different EVs tend to be different sizes. Now, but
I mean, like, I guess the other component here people would emphasize is, well, first of all, also EVs are super prevalent and common throughout the body, and so
yes.
They're getting used a whole lot, even though we don't have them all consistently the same size or whatnot. It might not be as big of a problem as we think it is. But
yeah, so there are a couple of different. We can talk about this for hours, but I'm mainly excited to see like the different forms of genetic and protein payloads, or sorry, the different carriers of genetic and protein payloads. AAV, genetic. LNP, mainly genetic. But things like exosomes and EVs can I know exosomes definitely can, but EVs, I think that they can also carry protein therapeutics. Maybe not. I don't know. What is this little thing? Is that a protein in there? I don't know. Okay. Anyways, it's a different form. I'm not super familiar with like the actual therapeutic advantages. I'm thinking though that EVs definitely overcome the immunogenicity, so allowing for redosing and probably simpler, like, I'm not an AAV guy either, so just forgive me. But I'm wondering, like, the big question that I would have is, like, can you get bigger payloads and different types of payloads, like proteins?
With EVs? Oh yeah, you can kind of throw in whatever you like. So it can be proteins, it can be mRNA, it can be
right. Yeah, and then the other thing would be like stability and circulation would be a big thing for me too. Like, how long do these things last? They pop pretty quickly. But anyways, that was the fourth and final emerging from stealth company, Spot Biosystems.
Right on. Shall we move on to the final signal of the day? Just a company that just keeps popping up. They are on a tear. Shall we move on to them, Jeff? Our favorite, Scribe Therapeutics.
Yeah, what'd they do? What did you do, Scribe? Not again.
Scribe has been on an absolute rip, ladies and gentlemen. They have raised a boatload of money. They are entering the clinic. They have a really cool therapeutic approach. They are spun out of Jennifer Doudna's lab, and they are focused on the epigenetic profiles of genes as opposed to editing the gene itself. And the signal that has them back in the news once again is they raised an additional $25 million through a non-dilutive grant to go after their preclinical programs, which is absolutely a nice chunk of change to get. So I guess like we could jump into like, yeah, what are the two preclinical programs here that have gotten the low density?
Interesting. Okay. So they're currently a lead collaborator on a clinical trial. They're not the lead sponsor. They're collaborating with Monash University in a phase 1 for patients with elevated LDL. Nice. So like high cholesterol. I'm wondering how that works. I don't want to dive into it. We don't have a whole lot of time, but yeah, this is, again, there's got to be something that I'm missing here with the epigenetic approach versus the genetic approach. Like, what is the purpose of doing epigenetic? I get that it's transient. I get it. But then just use an ASO. Like, use an antisense oligonucleotide. Like, what is the purpose of doing epigenetics? If you want transient like gene expression reduction, just use we have therapeutics to do that. They exist. So I don't know. Obviously, I'm wrong. Again, that's why he's wrong, because Jennifer Doudna's involved. So like, I'm obviously not, I'm missing something nuanced. But
who's the CEO at Scribe? I think it's another guy called Ben. Yes, it is Ben Oakes. Let's go.
Mm-hmm.
Ben Oakes.
He's probably a dork like you.
Shut up, Jeff. You're the one with a PhD.
I know one Ben. He's a huge dork.
There are more. There are many more.
So yeah, I mean, STX. I wonder if this is like if one of their STX 1150 is being used in the clinical trial. These are two different, more preclinical programs. I need to look into this and figure out what is the therapeutic advantage of using a epigenetic silencing therapeutic versus like just an mRNA degrading therapeutic. They both achieve the same thing: transient reduction in gene expression. But one of them just seems way more complicated, i.e. Scribe Therapeutics' approach. Just seems more complicated with more moving parts.
Well, I mean, with an ASO though, right? Isn't this something you have to keep dosing in?
Well, yeah, but like you get this weird like paradox where if the pitch for Scribe is it's not permanent, it's transient, right?
Yeah.
Then maybe that's I'm having a hard time finding the words clearly, but there's definitely like a paradox there where if you say that the therapeutic is meant to be transient, wouldn't that imply redosing?
Potentially.
Or is it just like, well, our therapeutic lasts longer?
Chris Spivey says epigenetic approaches offer three primary advantages over antisense ASOs: permanent therapeutic effects via single dose, the ability to activate gene expression, and it cut you off, Chris. Dang.
But I have a problem here though.
Okay.
Permanent therapeutic effects via a single dose. I don't think it's supposed to be permanent. If you wanted a permanent effect, then just cut it. Like, just turn it off. You can just cut.
Yeah, but that's I kind of get the idea. It's like instead of I think it's
continue the thought. No. Because you say if you don't want it to be so as dramatic as like cutting the genome, and it's the same effect. You're silencing the gene. It's the same.
Sure, the effect might be the same, but I would say it's probably somewhat similar to building out a mural by pulling out all the bricks and then putting in different colored bricks versus just painting on the mural. Just painting on the brick wall.
Here we go. This is going to be brutal, but here we go. It's the difference between me kidnapping your kid and locking him in a room forever or just killing your kid.
Okay. Now we're going to get
there's no difference. Like, according to you, my kid is gone forever, right? But it sounds better if you've got
to find less morbid examples.
Fine. It's kind of like
never having you to my house.
I have completely eliminated like any pizzeria ever. I've blown up the pizzerias. You can no longer have pizza, right? Versus I just don't allow you to order pizza ever again. Yeah, there you go. You're right.
Joke's on you.
You're right.
We're not getting monetized.
Yeah, we're already demonetized. So it's kind of like there's effectively no difference between transcriptionally repressing a gene or just cutting the gene out. There's no difference. You're going to lose expression either way forever. So I don't know. Maybe it's like there's less risk of mutational insertional mutagenesis, maybe.
Yeah, I would say it's the difference between a pizzeria getting demolished versus just closing the pizzeria down. And then if you ever want pizza again, you can just reopen it.
How? I'm not going to get into this. There's no mechanism within which you can reactivate gene expression here. And Chris's comment was permanent transcriptional repression.
No, it's permanent, but then you could go back in and put that in. You could put that epigenetic signature back on.
How?
Don't know.
Exactly.
That's for someone else.
You would need
figure
to
out down
have
the
some
road,
kind of like kill switch.
It's going to be way less intensive than rebuilding a pizzeria from the ground up versus just hiring a couple dudes to go in and start making pizzas in a closed down pizza restaurant.
We should move on.
Jeff, this is a recurring theme. You're like, Ben, you're stupid. And then 3 months later, you're like, well, you might have been right.
No, I'm saying, top of my head, it seems like there's something more nuanced that I'm not missing. That's, for me, it's like, well, what's the point? There's got to be. Their Scribe is going crazy right now. People are giving him money like left and right. So I'm obviously wrong. It's just like my Oonga Boonga brain is like, just use an ASO. What's the big deal?
Yeah.
So, anyways, okay. We good to wrap this thing up, Ben, or what?
I think so. All right, ladies and gentlemen, thank you so much for coming to the Biotech Voyager. We hope that you enjoyed our conversation today. We were able to have a great chat with the folks from Base Immune and the folks from Copra Bio. We will be cutting out clips from that soon, so take a look. If you aren't subscribed to the Biotech Voyager, please consider subscribing.
You're dead to me.
That'd be great. Yeah, we're trying to grow here. Excellent. All right. Well, ladies and gentlemen, thanks once again for coming. My name is Ben. I'm joined by Jeff.
It's me, it's Jeff.
My name is Jeff. My name is Jeff.
Excellent.
All right, ladies and gentlemen, thanks so much for coming. We'll see you later. Cheers.
Bing, bing, bing.